Hi, dear students. Today I wanted to discuss about the topic of cell cycle and cell division. So in this chapter, we are going to study about the definition of cell cycle. So the various steps involved in the cell cycle and cell division we are also going to discuss about. The types of cell division are the direct cell division or A mitosis, which you notice in case of the kingdom Monera. Example is binary fission, budding. These are all examples for amitotic cell division, and you notice this in kingdom Monera. Budding you can also notice in case of yeast. That is also a amitotic cell division. The other types of cell divisions are mitotic cell division, which you notice in case of the somatic cells. So the mitotic cell division is also called as equational cell division. And the third type that you are going to uh, study in this chapter is also going to be the meiotic cell division, which takes place in the reproductive cells or germinal cells, we call it as. And the type of cell division is also called as reductional cell division. The reason being the ploidy level of the parent cell in the newly formed daughter cell, it gets reduced. So from diploid, it becomes haploid. That is the reason meiotic cell division is also called as reductional cell division. So let us proceed in studying one by one each and every uh, concept that we have for this chapter. But before that, remember to like and subscribe for this channel, and also remember to press the bell icon to receive the regular notifications. If you have any doubts or clarification, you can send it through comments, and through the comments I'll be clearing it. So let us proceed with the chapter now. See the cell cycle, the definition of it. It is the sequence of events in which a cell duplicates its genome and synthesizes the other constituents of the cell to finally divide into two daughter cells. It is termed as cell cycle. So cell cycle is a sequence of events in which there is a duplication of genome, synthesis of other constituents of the cell, and finally it divides into two daughter cells. This entire sequence of events is termed as cell cycle. So that is what is the cell cycle. Now, what is genome? Genome is the sum total of genes in a haploid set of chromosomes. We call it as genome. So this genome replicates. So genome is the sum total of genes present in a haploid set of chromosomes. We call it as genome. Then the cell cycle, if you look into it, it is divided into two basic phases, interphase and M phase. So interphase is also called as preparatory stage, and M phase is mitotic phase. So the phases of cell cycle are interphase and M phase. So the mitotic phase involves two important steps, that is karyokinesis and cytokinesis. Karyokinesis implies it is the division of nucleus. Cytokinesis implies it is the division of the cytoplasm. So the nuclear division is followed by the cytoplasmic division in the M phase or mitotic phase. Now, in the cell cycle, the interphase stage is of long duration of time. It has various stages like G1 phase, S phase, and G2 phase. G1 phase is nothing but the gap 1 phase. S phase is the synthesis phase, and G2 phase is called as gap 2 phase. Sometimes some of the cells they also show the G0 phase or quiescent stage, during which even though the cell is metabolically active, they do not uh, get involved into the cell division. Still the cell requires a cell division to happen. So that G0 phase or quiescent stage is noticed in some of the cells. So the interphase is majorly made up of three phases: the G1 phase or gap 1 phase, S phase or synthesis phase, G2 phase or gap 2 phase. Now the mitotic phase, it has the karyokinesis or nuclear division. During this process, the cell division stages you can notice like prophase, metaphase, anaphase, and telophase. So this is followed by cytokinesis, that is division of cytoplasm. So mitotic phase involves the karyokinesis or nuclear division, during which the cell division stages you can notice, that is prophase, metaphase, anaphase, and telophase. And there is also division of cytoplasm, which we call it as cytokinesis. So the mitotic phase overall includes this. Now, the eukaryotic cell cycle, if you look into a typical eukaryotic cell cycle, it is given, the example given for this is human cells in culture. So they have noticed that for every cell division to happen in a human cell, so when I'm talking about cell division, I'm talking about mitotic cell division. For every mitotic cell division to happen in a human cell, it takes 24 hours. Of this 24 hours, 23 hours are required for the interphase stage, and they utilize only 1 hour for the mitotic phase. So the human cell requires cell division, it lasts only for about an hour. So the interphase lasts more than 95% of the duration of the cell cycle. It takes up a major role. So you can just assume that it requires 23 hours of preparation for 1 hour of cell division. So that is the importance of interface. So interface is not what a resting phase. It is rather a preparatory phase during which the cell gets prepared for entering into the cell division. The duration of the cell cycle, it can vary from organism to organism and also from cell to cell type. Now, in case of a yeast, you can see the cell cycle takes place within 90 minutes. That is, both interphase and cell division takes place within 90 minutes. But in human beings, it is 24 hours, of which 23 hours is for the preparatory phase and 1 hour it takes for the cell division. Now, if you look into this diagram is important because you will be uh, asked in the examination. This is the diagram that they have given in your textbook, and you can notice certain things like the at the beginning of the cell division, that is the cell cycle G1 phase, there is only one cell. At the end of the cell cycle, that is after cytokinesis, you can notice two daughter cells have been formed. So this cell cycle is one which is quite easy. I generally call it as pizza diagram because you find various slices, but you should also notice the duration of time is represented by this various sectors. G1 takes lesser duration when compared to S phase. S phase takes longer duration than G1 and G2 or M phase. So let us proceed with this cell cycle. So you notice that the cell in the it enters into the cell cycle, that is the interphase stage, G1 phase, gap 1 phase, during which the constituents of the cell increases, the nucleus enlarges, and it gets prepared for the uh, DNA synthesis to take place, but it does not bring the DNA synthesis. It gets prepared for that synthesis of DNA to take place. So there is the cell increases in volume and the nucleus enlarges. So these are the stages that you can see in the G1 stage. S phase or synthesis phase, as the name indicates, here the DNA doubles up. The DNA material doubles up. That is, they undergo replication to form two identical copies. But the number of chromosomes remain the same. The number of chromosomes are not going to double up. Only the DNA content is doubled up. All these are for the cell division to take place. These are preparation stage. So the synthesis phase, there is replication of DNA and doubling up of the DNA material, but the chromosome numbers remains the same. They do not double up. That you have to remember about. In the G2 phase, it is the growth of the cell further takes place, and you notice that there is protein synthesis which are required, of proteins which are required for cell division. That takes place in G2 phase. Then now they enter into the M phase, the mitotic phase, which is divided into two stages. That is the karyokinesis and cytokinesis. The karyokinesis has various stages, substages, that is prophase, metaphase, anaphase, and telophase. So the nucleus completely divides into two parts in the telophase. Now that is followed by cytokinesis or cell division. So the cytoplasmic division, cytokinesis takes place and the cytoplasm divides, and now you find that there is formation of two daughter cells. Both the daughter cells will have the same ploidy level. See, the parent cell had, just assume they had 46 chromosomes. The newly formed daughter cells will also be having 46 chromosomes. So 2n equals 46, and even the daughter cells 2n equals 46. So this implies that there is no change in ploidy level. So since the number of chromosomes have not changed and they have remained the same, so this type of cell division is also called as mitotic cell division, equational cell division. So mitotic cell division is also called as equational cell division. See, in the interface, what you can notice is it involves a series of changes that prepares the cell for division. It involves the period of cell growth and cell division in an orderly manner. It is divided into three phases. See, the interface is going to have a series of changes which prepares the cell for division. It is involving the period of cell growth and cell division in an orderly manner. So it is divided into three phases, that is G1 phase, S phase, and G2 phase. In G1 phase, it corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. So during G1 phase, the cell is metabolically active and continuously grows, but there is no replication of DNA. It just initiates the DNA replication at this stage. So, and you should also remember G1 phase is the interval between mitosis and synthesis phase. It is in between mitosis and synthesis phase. So sometimes G1 phase is also called as post-mitotic phase. So G1 phase is also called as post-mitotic phase, and G2 phase is called as pre-mitotic phase, that is just before mitosis. So the S phase, the next stage is the S phase or synthesis phase. In this phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. So there are two things are happening here. DNA replication begins in the nucleus, and in animal cell, the centriole duplicates in the cytoplasm. Centriole is absent in plant cell. The amount of DNA doubles up, but the chromosome number remains the same. So in the G2 phase, that is the third stage, which we also call it as pre-mitotic phase. So G2 phase is present between S and M phase. So G2 phase, the proteins are synthesized in preparation for mitosis while cell division continues. So the proteins are synthesized in preparation for mitosis while cell growth continues. That is what you have to remember about. Then G0 phase or quiescent phase, what you notice here is the cells in this stage, they remain metabolically active, but they no longer proliferate. That is, they no longer undergo cell division unless called on to do so. Whenever the requirement arises in the organism, they undergo cell division. So the G0 phase or quiescent phase, in this stage, the cells are remain metabolically active, but they do not proliferate or they do not undergo cell division unless required by the organism. So that is what you have to remember about. The M phase or mitosis. It is a process during which the number of chromosomes in the parent and progeny cells is the same. When we are talking about progeny, I am talking about offspring or the daughter cells, you can also call it as. So where the number of chromosomes in the parent and progeny cells or daughter cells remains the same. So since the number of chromosomes remains the same, it is also called as equational division. Example I told you, when a human cell is undergoing mitotic cell division, so the parent cell will be having 46 chromosomes, that is 2n equals 46, that is diploid number of chromosomes. So when they undergo mitotic cell division, they are going to form two cells, two progeny cells or two daughter cells, and both of them and each one of them will be having 46 chromosomes. So there is no change in the ploidy level of the chromosomes or number of chromosomes. That is the reason we call mitosis as an equational cell division. The process of mitosis, it keeps the chromosomes number equal in daughter as well as the parental cell. So mitosis usually takes place in non-reproductive cells or vegetative cells, or we call it as somatic cells. So they take place in non-reproductive cells or vegetative cells or somatic cells. So the mitosis involves four stages. They are prophase, metaphase, anaphase, and telophase. Also, you should remember mitosis involves two stages, karyokinesis and cytokinesis. Karyokinesis is the division of nucleus which happens by the prophase, metaphase, anaphase, and telophase. And cytokinesis is the division of cytoplasm by the formation of cross walls or cell walls. So there is the formation of two daughter cells. So the division of cytoplasm, we call it as cytokinesis. So both the steps are involved in mitosis. If we get into the prophase of mitosis, what you can see, the key attributes here is there is going to be the disappearance of nucleus and nucleolus and the nuclear envelope along with cell organelles, and there might be the initiation, that is beginning of the assembly of mitotic spindle, microtubules, and proteinaceous components of the cell cytoplasm, which help in the mitotic process. So here you can also notice within the nucleus, the chromatin material or chromosomal material, which is thread-like, they becomes untangled and they are going to condense to form chromosome material. So the chromosomal material condenses to form compact mitotic chromosomes. So chromosomes are composed of two chromatids attached together at the centromere. That is we call it as sister chromatids. So the chromosomes are having four arms there totally. That is the chromosomes are seen to be composed of two chromatids. So which are held together at the centromere. So are bivalent chromosomes they are. So the other steps that are taking place in the prophase stage is initiation of the assembly of mitotic spindle, microtubules, the proteinaceous components of the cell cytoplasm. So all this initiation happens so that they can be utilized during the further cell division stages. At the end of prophase, in the cell, you can notice that the Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope, they disintegrate or they disappear or disintegrate. They are not seen anymore. So they disintegrate. So you can see the stage, the early prophase, you can see there is the chromatin material or chromosomal material is highly uh, uncoiled. They later become coiled to form condensed structure. So in late prophase, you can see that there is a distinct chromosomal material with centromere. So each chromosome has two chromatids, which are nothing but sister chromatids, and they have a centromere. So this you can notice, and there in the late prophase you can also notice the disappearance of nuclear membrane, nucleolus, so the cytoplasmic organelles like endoplasmic reticulum, Golgi complex, they all disappear during the late prophase. So metaphase stage, you should remember that the condensation of chromosomes is completed. So metaphase stage is the ideal stage to study the morphology of chromosome. If you want to study the structure of a chromosome, so metaphase chromosome, if you choose, you can study it in very detailed manner. So in the metaphase chromosome of mitosis, you can see two sister chromatids which are held together by the centromeres. And the centromeres, they have small disc-shaped structures present at the centromeres, which are called as kinetochores. So they are present on both the sides of the centromere, the kinetochores. So they serve So what is the purpose of kinetochores? They serve as the site of attachment of spindle fibers. See, the spindle fibers are attached to the kinetochores from the opposing poles. So they are formed by the spindle fibers to the chromosomes that are moved into the position at the center of the cells. So the kinetochore structure serves as the sites of attachment of spindle fibers to the chromosomes, which are moved into position at the center of the cell. So in the metaphase, you can notice that the all the chromosomes are at the center of the cell, so which we call it as equatorial plate. So they are at the center of the cells. So they get arranged there, and you find the spindle fibers of each chromosomes. The opposite poles they are attached to one side of the kinetochore. So there is a attachment of spindle fiber to one end of the centriole, the centromere of the kinetochore. The other end of the same centromere is attached to the opposing spindle fiber. So you can notice that. The next is metaphase is characterized by all the chromosomes which come to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. So you have two sister chromatids. One sister chromatid is connected to the spindle fibers by its kinetochore. Another spindle fiber, another sister chromatid is attached uh, to its kinetochore by spindle fibers from the opposite poles. So the chromosomal material condenses to form compact chromosomes that get aligned in the middle of nucleus at equatorial plate. That is the important things. The chromosomes are arranged at the center of the nucleus on the equatorial plate. They are arranged, all the chromosomes. So that is the important feature of metaphase and identifying feature of metaphase. Now, if you look into this diagram, you can notice that the transition to metaphase. So in case of animal cells, you notice that the centrioles would have formed the asters and spindle fibers, and they are all attaching to the centromeres. So in the metaphase stage, they realign, all the chromosomes realign at the central equatorial plate, and you can notice there are two sister chromatids in each chromosome. So one of the centromere of one sister chromatid, so at the kinetochore, it is attached to the spindle fibers at its kinetochore, one sister chromatid. The other sister chromatid's kinetochore is attached to the opposing spindle fiber. So they are having two strings attached to the spindle fibers attached in opposite poles for the same centromere. So later it would be leading to it is similar to the tug of war. So when the chromosomes are pulled from both the ends by the condensation of the spindle fibers, the chromosome breaks at the center, the centromere splits, and the sister chromatids they get separated. One sister chromatid moves to the one pole, the other sister chromatid moves to the other pole. So what you can notice here is the bivalent chromosome becoming univalent by the splitting up of centromere. That takes place in the anaphase stage. But what you have to remember in metaphase stage is all the central chromosomes are arranged at the center of the cell on the equatorial plate, and they are attached by the spindle fibers at its kinetochore. So the centromere in the uh, chromosome, it has kinetochore at both the poles, facing both the poles which are attached by the spindle fibers, and they create a pulling force similar to a tug of war, which is being tugged from both the ends. See, anaphase is a stage in which the chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as chromosomes of the future nuclei, so they begin their migration towards the opposite poles. So the sister chromatids they break at the centromere and they move towards the opposite poles. Uh, so that is what is happening here in the anaphase stage. As each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole. you can notice that the centromere of each chromosome is towards the pole and hence at the leading edge, with the arms of the chromosomes trailing behind because they are pulling them by the centromere. So centromere is going to be facing ahead and the chromosomal arms will be behind. So that is what you have to remember about anaphase. The key events in the anaphase are the centromere split and chromatids, they separate. The chromatids move to opposite poles. That is the key features or key events that is taking place in anaphase. If you look into the diagram, so you can see that the sister chromatids have split at the centromere, and by the condensation of the spindle fibers, they are pulled to their respective poles. And they are first, the centromere is facing towards the pole, and the chromosomal arms are lagging behind. In both the ends, you can see that centromere will be leading ahead, and the chromosomal arms are lagging behind. So they are pulled towards the opposite pole. So this sort of arrangement you notice in anaphase. So in telophase, what happens is the nucleus they gets reorganized. Nuclear membrane reappears, nucleolus reappears, and the coiled chromosomes they are going to get uncoiled. So the compact chromosomes they are going to become uncoiled. So you notice that in the telophase. See, the chromosomes that have reached their respective poles, they decondense, that is highly uncoiled, and lose their individuality. The individual chromosomes can no longer be seen, and you can see them as chromosomal material or chromatin material, which collects as a mass in the two poles. So the key events during telophase are the chromosomes cluster at opposite spindle poles and their identity is lost as a discrete element. They are not seen as a discrete element chromosomes. They are seen as the chromatin material or chromosomal material. The nuclear envelope, they assemble around the chromosomal clusters. The nucleolus, Golgi complex, and the endoplasmic reticulum, they are going to reform. They are going to appear back again. They are going to be resynthesized again in the telophase stage. So now there is a complete division of nucleus. Now after telophase, they immediately enter into cytokinesis, that is division of cytoplasm. So the after cytokinesis, they enter again into interphase. So the cycle continues. So karyokinesis is the division of nucleus during mitosis or meiosis, which is followed by cytokinesis. So cytokinesis, as I've already told you, it is the division of cytoplasm of a cell. In an animal cell, uh, the cytokinesis is achieved by the appearance of a furrow in the plasma membrane. You can notice in this diagram there is an appearance of furrow in the plasma membrane. This is taking place, furrow in the plasma membrane, which is going to meet at the center, and there is going to be a cross wall formed by this furrow, which deepens, and they are going to form a cross wall. So that is what you notice in case of an animal cell, the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, which dividing the cell cytoplasm into two. In plant cells, wall formation starts in the center of the cell and grows outward. So the plant cells, the wall formation is uh, starting from the center of the cell and they are going to grow outwards to meet the existing lateral walls and they form a cell plate. So they are in the center as a cell plate. So they grow outwards or towards the sides to meet the existing lateral walls and they form a continuous cell plate which divides the cytoplasm. So at the time of cytoplasmic division, the organelles like mitochondria and plastids, they get distributed between the two daughter cells. So you find the mitochondria and plastids are being distributed amidst the two newly formed daughter cells. When in certain cases the karyokinesis is not followed by cytokinesis, you notice a multinucleate condition is noticed. Even in fungi, you'll be later studying about syncytial hyphae or and septate hyphae, non-septate or syncytial hyphae. So what is this condition is if karyokinesis is not followed by cytokinesis, there would be a multinucleate condition. This we call it as syncytium. Example for the syncytium is liquid endosperm in coconut is a very good example for syncytium. The milk of the coconut or the coconut water, tender coconut water, that is an example for liquid endosperm. Why they are liquid endosperm is they have free nuclear nature because there is no formation of the uh, cross walls. Why there is no formation of cross walls? Because karyokinesis is not followed by cytokinesis. So one of the examples for karyokinesis happening but absence of cytokinesis is the syncytium, which is noticed in case of liquid endosperm in coconut. Significance of mitosis. So mitosis, they result in the formation of diploid genetically identical daughter cells. So there is no change because there is no gene recombination or crossing over. There is nothing at all happening here. So they result in the formation of diploid genetically identical daughter cells. The growth of the body takes place by mitosis. So growth, whenever we say, we call it as cell division, cell enlargement, and cell maturation. All three stages are involved in growth. Growth is an irreversible increase in mass or volume of an organism with reference to time. Irreversible increase in size or mass of an organism with respect to time. So growth of the body takes place by the mitotic cell division. Then cells repair and replacement of worn out tissues is by mitotic cell division. You have an injury in your hand, the new skin is going to be formed, or the healing process involves the formation of new skin, which is by the mitotic cell division. The maintenance of nucleocytoplasmic ratio, whenever there is a difference in this nucleocytoplasmic ratio, they undergo cell division. So mitosis helps in maintaining of a constant nucleocytoplasmic ratio. The vegetative reproduction in plants, it takes place by mitotic cell division. So these are the significance of mitotic cell division. So you should remember in this topic the various things that we have studied is we have began with the definition of cell cycle, which is a sequence of events in which there is the doubling up of genome, then there is also doubling up of other components of the cell, and finally there is division of cell into two progeny cells or two daughter cells. So that entire sequence of events we call it as cell cycle. So cell cycle, they have interphase stage and M phase. Interphase stage is subdivided into the G1 phase, S phase, G2 phase, and sometimes in some of the cells they also have G0 phase or quiescent phase. Then the M phase is having or M phase or mitotic phase, it has two stages, karyokinesis and cytokinesis. So karyokinesis is division of nucleus, cytokinesis is division of cytoplasm. So these stages are involved in cytokinesis, and we also studied about the interface in detail. We studied about the various stages. Then we studied about mitotic cell division. Especially we began with karyokinesis, that is prophase, anaphase, metaphase, anaphase, and telophase. What you can remember about is which is unique to mitotic cell division is in metaphase, uh, the spindle fibers are attached to the kinetocosi, opposite kinetocosi of the centromere, and in the anaphase, the centromere splits at the center and the sister chromatids separate and they start functioning as chromosomes. So the splitting up of centromere takes place in the anaphase stage because the spindle fibers are attached to the kinetocosi of the opposing side of the centromeres. As a result of it, it is like a tug of war, and the centromere at the centromere they break, and the condensation of the spindle fibers leads to the pulling up of chromosomes towards the opposite poles, which you can notice in the anaphase stage. So that is an important sequence that you should remember. So the bivalent chromosomes have become univalent chromosome. That is also one of the point that you have to notice. So that might be absent in meiosis 1, but in meiosis 2 you can notice the same stages. So meiosis 2 is also called as haploid mitosis. So these are the points that we have discussed for today's uh, session. So if you have liked this discussion, if you have liked this video, you can share it with your friends, and also remember to subscribe for this channel, and also press on the bell icon to receive regular notifications. Thank you. So best wishes for your academic year ahead.